Hello everyone, I am Dr. Reha Rakholia. Welcome to my channel where today we'll explore the culture system of human embryo. Feel free to ask questions in the comment and subscribe for more contents in infertility. Let's dive in. Culture system of human embryo. Culture system of human embryo is a complex task. Okay, it's a complex task. It required training of embryologist. It required training of embryologist, and it needs human embryo culture needs stable environment, and it is beyond media formulation. It does not only include culture media. Whenever we talk about culture, we think that it talks about culture media, but that's not true. Okay, what all it contains is culture system contain media. First of all, it contains media. Second is incubation chamber. Third is macromolecule. Okay, macromolecule like amino acid and hyaluronin. Fourth is gas phase. Like there is a CO2 incubator and there is trigas incubator. So it is known that 5% O2 is good for the embryo rather than 20% of O2. Okay. So gas phase is also a part of culture system. Fifth is air quality is also a part of culture system. Sixth is culture vessel. In what we are culturing? Culture vessel. Seventh is media overlay. Oil overlay is there, no? Which we apply over the media. Like this is the media. This is oil overlay. Okay, oil overlay. So that substance don't get like volatile substance don't get evaporated. So we apply oil overlay, and it also prevent from increasing the osmolarity. Okay. Eighth is embryologists themselves. These 8 things include culture system. Okay. These 8 things include. As we have already talked about component of culture system, media, macromolecule, gas phase, type of medium overlay, culture vessels, incubation chamber, ambient air quality, embryologists themselves. See, this is culture media. This one is culture media. Okay, this is embryo in the culture media. This is oil overlay. These are macromolecules like albumin and hyaluronin. Okay. These are incubator, CO2 incubator and trigas incubator. Now stressors. Okay, we'll talk about stressors. Stressors in embryo culture. What are the stressors? First is O2 exposure, oxygen exposure. Okay. Second is, like I have already talked, that 5% oxygen is good and 20% oxygen is not that good. Okay, this is good, 5%. So we'll talk about it later. Then second is CO2 and pH change. CO2 and pH change, like if we'll do in and out of incubator, in and out, that the culture media in which there is embryo, if we'll regularly do in and out of incubator, then CO2 and pH changes will be there, which is a stressor for the embryo. Third is temperature shift. Okay, temperature shift. This is also a stressor for embryo. Fourth is physical stress from vigorous pipetting, physical stress. Fifth is accumulation of ammonia accumulation from amino acid. Okay. Sixth is, these are all stressors. Sixth is osmolarity. This will increase, as I have already told. It will increase if we won't use overlay media. It will increase. Then we'll talk about holistic analysis in IVF lab. First, we already know that earlier stages of development, particularly pre-compaction stages, are more susceptible to stressor. These are most susceptible. As we go from pre-compaction stage to compaction stage, stress will decrease. Susceptibility to stress will reduce. Stress will not decrease, but susceptibility to stress will reduce. Embryo won't become that susceptible to stress. Stress can interact negatively, creating adverse synergy. Then now, now we'll talk about holistic approach. Analysis of IVF laboratory. First is simplistic assumption. We think that things are very simple, but it is not. Like, we think if we'll change one part of something, like if we'll change culture media, that will mimic the laboratory in which results are like that, and so on. But it is not like that. Only culture media is not responsible for the result of given lab or clinic. QC, QA, significantly quality control. I'll talk about it in another lecture. QC, QA, quality control and quality assurance is it significantly impact on success of IVF. Okay, QC, QA, and second is health and diet. Third is, see, there is complexity in the treatment. What is this complexity in the treatment? First is stimulation regimen. Stimulation regimen decides the oocyte quality and embryo physiology and viability. Oocyte quality, embryo physiology and viability, and endometrial receptivity. Okay, stimulation rate. So it's complex procedure. All these contribute to the success of embryo. Then is laboratory component. What are laboratory component? Like culture system, embryologist, incubator, number of incubator, and air quality. These are lab component. Culture system, embryologist, number of incubators, and air quality. Then fifth is quality control. Fifth is quality control. Like, if some untested material is there, it is considered toxic unless proven otherwise. Okay, we have already discussed this. Simplistic assumption is only culture media is responsible, which is not. And now we'll talk about one cell mouse embryo assay. Okay, one cell mouse embryo assay. This is a pre-test. Okay, used to pre-screen every lot of tissue culture wear. Then 7th point is around 25% of material fails the one cell MIA test. Around 25% जो होता है fail हो जाता है MIA test में. Okay, mouse embryo assay. Then seventh is resource challenges and resource allocation. Okay, there are, first is lack of resources for quality control. Lack of resources for quality control. And second challenge is that media is considered a culprit, but most frequently labware is a culprit. Labwares are the culprit. ये नहीं है culprit. It is not the culprit. This is the culprit. Okay. Then, okay. See, it is explained nicely here. Patient's etiology, genetic, diet, stimulation, This decide the oocyte quality and impact on the endometrial receptivity and embryo transfer and luteal support. These all are responsible for the outcome. And in lab, what all are there? Number of embryology and training level, number of incubator, air quality, and culture system. In culture system, there is tissue culture where contact supply, culture media, overlay, oil overlay, number of embryos per drop, number of embryos per drop, and gas phase. Okay, number of embryos per drop and gas phase. Okay, then this diagram shows that relative impact of chemical and physical stress of the embryo decreases as we go from the pre-compaction to post-compaction. Okay, sensitivity to stress decreases. The blastocyst is least affected by chemical and physical stress. This one is least affected by chemical and then maintaining gamete viability. Maintaining gamete viability. See, there are three factors. First is patient factor. Second is endometrial receptivity. And third is ovarian stimulation. These all decide the gamete viability. Okay, patient factor like diet, genetic, etiology. Endometrial receptivity, we have already read that endometrial receptivity is decided by the ovarian stimulation also. Okay, other factor also, but ovarian stimulation also. And ovarian stimulation also affect the quality of embryo and endometrial receptivity. Okay, gamete quality and endometrial receptivity. Then we need to have a communication. We need to have a communication. See, communicative pathway for the gamete viability. What is communicative pathway means? There should be communication between physician and embryologist. Okay. Then the human embryos are considered shows plasticity. Okay. It shows human embryo in culture shows plasticity. What does that mean? It can develop under various culture condition. Then if we give suboptimal condition, this may impair viability and pregnancy outcome. Okay, viability and pregnancy outcome may get impaired because of the suboptimal condition. And see, implantation to evaluate lab performance. Implantation and FHR, fetal heart rate, is an important parameter. But true efficacy of IVF lab, implantation and fetal heart rate is an important parameter. But if we'll have to know the true efficacy of IVF lab, that is decided by live birth rate and cumulative birth rate. Okay. Now, even if the embryo is going to till blastocyst stage, that does not mean that embryo, that does not ensure viability. Maybe it has that does not ensure viability. Maybe it has previously had so much stress that growth development can stop. It does not ensure viability if it's developed till the blastocyst stage. Okay, then key parameter eruption. Advances in embryo culture and selection. What are the advances? See, nowadays we do SET, single embryo transfer. We try to do single embryo transfers. Why? How was that possible? Because of the enhanced culture enhanced culture system, which must provide high quality embryo and also impact cumulative pregnancy rate. Okay, what is cumulative pregnancy rate? Cumulative pregnancy rate is the rate of the pregnancy per egg retrieval. Okay, it's important to know the denominator. It's per egg retrieval. It is per egg retrieval. Then this culture system also has effect on cumulative pregnancy rate. Okay, now first we learn about culture system. Culture system has improved, that's why we are able to do set. Second is cryopreservation. Cryopreservation program has improved. Why is it important? Because the remaining embryo needs to remain viable after the cryopreservation for later pregnancy. Okay? If first transfer does not result in the pregnancy, so we need to have a good cryopreservation program. Third is high quality embryo. How we are able to develop high quality embryo? First, I have already said, because of culture system has become good, enhanced culture system. Second is because we have a better embryo selection method. Because of better embryo selection method, we are able to see the high quality embryo. Okay. And advances in culture system. What are the advances in culture system? First is culture media. There are different type of culture media nowadays. Like if we want like hyaluronin is added. Hyaluronin is added to increase the survival after cryopreservation. Second is there is improvement in culture cryopreservation. Third is nowadays we are culturing till the blastocyst, and there are various advantage of culturing till the blastocyst. Okay, and genetic screening also has a advantage. We can have a good quality embryo because of genetic screening, PGTA, and we'll transfer it first. So SET is possible. Okay, SET is nowadays a standard of care to avoid multiple birth. Now we'll talk about embryo and maternal physiology. See, it's important to understand cell physiology. Okay, because there are energy preferences in the pre-compaction cells and post-compaction cells. Okay, so pre-implantation human embryo is dynamic, changing from the quiescent pronucleated oocyte to a highly active blastocyst in 4 days. Okay, so cell physiology is important to understand. From quiescent pronucleated stage, it is changing into highly active blastocyst stage. Okay. Second is early stages prefer pyruvate, and late stages prefer glucose. How will we remember this? जब हमारा शुरू में प्यार होता है, पायरुवेट मीन्स प्यार. ओके, जब शुरू में प्यार होता है तो वो अर्ली में प्यार होता है ज्यादा. ओके, तो अर्ली इज पायरुवेट. शुरू में प्यार ज्यादा होता है तो अर्ली इज पायरुवेट, लेट इज glucose. In late stages we this embryo use glucose. Okay, Then importance of metabolic homeostasis. What is the importance of metabolic homeostasis? There are key development event. Okay, there are key development event like egg activation and regulation of methylation and acetylation. These are the key development event. We are talking about metabolic homeostasis. Okay, egg activation is there. Second is regulation in the methylation and acetylation. These key development event depends on the protein. So that's why protein is very important. Okay, second is in early stages, there is less biosynthetic activity. Biosynthetic activity is less, and but nuclear remodeling is there. Nuclear remodeling is present. Modeling is present means there is methylation and acetylation. This effect, methylation and acetylation, these affect DNA and protein synthesis. We are talking about early stages. Okay. Now, there should be balance between mitochondrial and cytoplasmic metabolism. There should be balance between mitochondrial and cytoplasmic. Now, like we talked before, that from pronucleated oocyte to which is quiescent to highly active blastocyst. Okay, what oocyte? It also prefer pyruvate, and it has a pronucleated low oxygen consumption. Okay, like M2 oocyte, this pronucleated oocyte also prefer pyruvate, and there is low oxygen consumption. Okay, there is increase in the energy demand. We'll talk about how there is increase in the energy demand. First is post compaction. There is embryonic genome activation. In this, there is transcription and protein synthesis. That's why there is increased demand. As we go from the early stages to the later stages, there is increased energy demand because of the embryonic genomic activation. Second is because of the cell multiplication. Okay? High energy demand will be there. Third is because of blastocele formation. High energy and glucose utilization will be there. Okay, now blastocyst stage metabolism. What will happen in blastocyst metabolism? What will happen is, first is high oxygen consumption. Second is there will be aerobic glycolysis. And what is aerobic glycolysis? This is high, it produces lactate. Okay, like Warburg effect. We usually say Warburg effect, no? Warburg effect. What happen is, as we know that in cancer cell, what normal cell does is it breaks down glucose. Glucose breakdown using O2. Using O2 in mitochondria to produce lots of energy. What is this aerobic glycolysis is? Lysis is, first of all, it is like in cancer cell, it happens in cytoplasm. Breakdown of glucose without using much oxygen. Okay, glucose breakdown without using much oxygen. Oxygen ज़्यादा use नहीं हो रही है, but there is glucose breakdown. It is less efficient. It is less efficient, but very fast. But fast. That's why there is accumulation of lactate. C3, increasing energy demand, we have already told. Cell multiplication, embryonic genome activation, and blastocell formation. Because then this high oxygen consumption, glucose utilization, and aerobic glycolysis, Wartburg, I have already discussed. Then pre and post compaction differences. What are pre and post compaction differences? See, in pre compaction, it resemble like pre compaction. It resemble like unicellular organism. Okay, unicellular organism. It has low oxygen consumption. Unicellular organism. First is low oxygen consumption. Second is prefer pyruvate. I told you ना तैयार कब होता है early stage में, तो pyruvate is in early stage. Then low biosynthetic activity. Now post compaction, high oxygen consumption. High oxygen consumption. This prefer glucose. In this, formation of transporting epithelium is there. Of transporting epithelium is there, and high biosynthetic activity. See, as we have already seen, biosynthetic activity, we have already discussed it. I'll talk about the amino acid requirement. In pre-compaction, I'll talk about it later. This genome dependency, pre-compaction is maternal genome, post-compaction is embryonic genome. Cell type, it is one cell type, it is two distinct cell type, inner cell mass and trophoectoderm post-compaction. Okay, ठीक है. Now see, there are oviduct fluid and uterine fluid. Oviduct will contain pyruvate, lactate, and oviduct will pyruvate, lactate, and low glucose. Okay, high pyruvate, high lactate, and low glucose. And uterine fluid will contain low pyruvate, low lactate, and high glucose. So it's kind there, like whatever there is requirement, they're changing. In vivo also, it's changing according to the requirement of the embryo. Okay. Now this is my way of learning the amino acids in pre-compaction stage. Amino acid in pre-compaction stage. Okay. See, alien, how is, can you see the alien? This is a picture mnemonic. This will tell me about the alanine. Then serine monk is there. This will tell me about the serine. Then spartate will tell me about the sparkling star. See, sparkling stars are there. Then glutamate, glowing moon is there. This will tell me about the glutamate. Then glycine is gliding swan. This swan is gliding here. Glycine, very nicely it is gliding. Swan is very nicely gliding. So I'll run gliding swan. Proline is pro surfer. This surfer is pro. Pro means very good. So proline I'll remember by pro surfer. Taurine is I'll remember by bull. Bull will tear you apart. Taurine. Okay. This way we'll remember the amino acids in the pre-compaction state. Now, nutrient preference and energy substrate. I have already told you that in oviduct there is high pyruvate and lactate and low glucose. In uterine fluid there is low pyruvate and lactate and high glucose. Then susceptibility of pre-implantation embryo to stress. Okay. First is early embryo will adapt to the environment, but at a cost. Okay, at a cost, because stress pathway disruption affects fetal growth. Okay, how? See, embryo initially may look normal. Embryo will be looking normal, but there is cellular disruption and it will become a non-viable embryo. First, it will look normal. During stress, it may look normal, but it will become non-viable and there will be cellular disruption. And implantation may occur even if that embryo is in stress or it has gone through stress. Implantation may occur, but there will be growth retardation. Okay, and stress also affect implantation and pregnancy rate. Okay, this are morphology alone cannot distinguish between viable and non-viable embryo. According to morphology only, we cannot say. We'll see the cell in the microscope and we'll say, okay, it is viable or non-viable. That is not possible. Alone cannot distinguish. And stress effect may not be evident until the blastocyst stage. Until the it may not be evident until the blastocyst stage. Now we'll talk about the cleavage stage as post-compaction embryo. Okay. Now, pre-compaction embryo, this struggle with stress and gene transcription. Pre-compaction has limited ability to transcript gene. What is transcript gene? Gene transcription. What does that mean? See, it is for a cellular function, this is the initial gene transcription is the initial step. Gene transcription is the information coded in DNA. Okay, what will happen? DNA information will code into mRNA. mRNA, then protein. mRNA will act as a template. It will form protein, which will do the cellular function. That means initial function is through gene transcription through the DNA. Okay, now what else? Cleavage stage relies on the maternal genetic material and protein. That's why the activity is capacity of it is restricted to adopt and regulate its internal processes. Okay. Next, we'll talk about, see, morphology alone cannot distinguish between the viable and non-viable embryo that we have already talked. Now, importance of culture conditions. Okay. Importance, what is there? It is very important to ensure normal cellular physiology. Okay. It is a key for embryo health. Not to ensure the normal physiological cellular physiology is the key for embryo health. And there are limitation in morphological assessment. Like morphology cannot assess everything about the embryo. So for that, we need advanced diagnostics. Okay. There is, see, composition of culture media, basically it's a crucial for embryo development. And basically there are two things, amino acids and macromolecule. Okay. Right media will enhance embryo development. If you use the right media, it will enhance the embryo development. Then role of amino acids. Role of amino acids. What are the various role of amino acids? Oviduct and uterine fluid contain high level of amino acids. Amino acids are essential for the embryo development. Oocyte and embryo have a specific transfer system for amino acids. See, roles are biosynthesis, synthetic precursor. It is the biosynthetic precursor, B is it acts as energy source. C is regulation of energy metabolism. D is osmolytes. It acts as buffer also. It acts as antioxidant. It acts as chelator, signaling, regulation of differentiation. So there are many roles of amino acid. Then role of importance of amino acids in oviduct and uterine fluid. Now there are two things. First is non-essential amino acid. What does it do in non-essential amino acid and glutamate? Non-essential amino acid and glutamate. What it does is increases the cleavage rate up to 8 cell stage. ये आपका प्री कंपेक्शन की बात हो रही है, ठीक है. We are talking about the pre-compaction here. Non-essential amino acid and glutamate increase the cleavage rate up to 8 cell. Where is it given? Up to 8 cell, non-essential amino acid and glutamate increase the cleavage rate. Okay. Now, what it does in post-compaction? Post-compaction, non-essential amino acid plus glutamate. First of all, it will help in the blastocell formation. Blastocell formation. Blastocell formation. Second of all, hatching. And what does essential amino acid does? In post-compaction, improve cleavage rate and inner cell mass development. Basically, amino acids improve embryo viability after the transfer. Okay. Now, non-essential amino acids can also act as a chelate. It can also it chelates the toxin. Okay. It also regulates the homeostasis before compaction. Homeostasis also it maintain homeostasis, chelate. It also acts as intracellular pH buffer. We are talking about the non-essential amino acid right now. Non-essential amino acids. All these actions it is doing. Okay. See, now we'll talk about the role of non-essential amino acid. We have already. It is a deuterionic nature, means it can act as both positive and negative charges, but overall it is neutral, means it maintain the, see, it has a positively charged amino group and a negatively charged carboxyl group, so it acts as a buffer. It helps in homeostasis, cellular homeostasis, particularly in early stage of embryo development. We have already discussed it is a strong pH buffer, it chelate toxins, and it regulate homeostasis. We are talking about the role of non-essential amino acids. Post-compaction changes. After compaction, there is a transporting epithelium. So embryo can regulate their internal environment better. They become less dependent on non-essential. After compaction, they become less dependent on non-essential amino acid for regulation of intracellular function. अब है re-evaluating amino acid classification. See, that that term non-essential and essential was for somatic cell. Okay, this non-essential and essential term is not for this embryo. It was for somatic cell. Okay. Then there is essential amino acid like methionine and cysteine, crucial during the cleavage stages. During cleavage stages, what methionine does is methylation pathway. It helps in the methylation pathway. And what this cysteine does is it helps in the antioxidants. It acts as a antioxidants. Now we'll talk about the embryo viability. See, amino acid exposure and embryo viability. What is the impact of what is the impact of amino acid deprivation on the culture media? For even if we'll deprive amino acid for 5 minutes, even if we'll deprive amino acid for 5 minutes from the culture media, it will impair development. And to restore this takes several hours. Okay, transient exposure 5 minutes also will impair the development, and it takes to replenish several hours. Then what is the clinical implication? Clinical implication is like we handle oocytes. So during oocyte collection and manipulation like ICSI, the holding media should contain the amino acid. Holding media should contain amino acid. Okay, holding media should contain amino acid. Now we'll talk about post-compaction changes. Post-compaction is less dependent on amino acid. Post-compaction is less dependent on amino acid, but amino acids are important in every stage. Okay. So what it helps in, it also helps in aligning gene expression. Also help in aligning gene expression. Okay. It also help in aligning gene expression and decreases the intracellular stress. Cell के अंदर stress है, it decreases that. So it supports in development. Post compaction जो है better internal environment regulate करता है. Then pre compaction it. Okay, now we have done this. We have already read that gene expression को ये amino acid in culture align करता है. Now macromolecule in embryo culture. Okay. There are two macromolecules basically, albumin and hyaluronan. Now albumin. Historically, we used to use serum albumin. But now it is avoided due to detrimental effect. It is because it is contaminated with fatty acid and other small molecule, and there is variability between the batches and sources. And there are embryotropic factors and contaminants like diethyl hexyl thalate, okay, and monoethyl hexyl thalate and polybrominated diphenyl ethers. Okay, and there are variable level of carbohydrate, amino acid, transition metals, and growth factors. So serum albumin is not used now. Now what we use is recombinant human albumin. Okay, it is it standardizes media formulation. I mean, it is standard. There is no variability, and it eliminates the issue inherent in blood derived product, and also because it is not blood derived, it is recombinant, and also if it is effective in supporting fertilization and development. And before it was costly, that's why we didn't use it. Recombinant human albumin we used to avoid because of its cost, but now costs are decreasing, so we can use recombinant albumin, human albumin, instead of serum albumin. Second is hyaluronan. Okay, it improves the culture system and it has a clinical benefit in cryopreservation. Okay, the survival rate increases, and trial also showed that pregnancy increases, implantation increases, and live birth rate increases after the hyaluronan used in the culture media. It has been, I've already discussed, it has benefits in cryo survival. It has benefits in, and this is, we are talking about hyaluronan. In trials, we have seen that there is increased pregnancy rate, implantation rate, and live birth rate. And there is target group benefiting from hyaluronan, that is above 35-year-old, poor quality embryo, and previous implantation failure. These three groups benefits from hyaluronan more. Okay, there is more benefit for these three groups. Then In vitro artifact and ammonium production. In vitro artifacts. First, there is in vitro means non-physiological environment. Okay. In vitro means a non-physiological environment, and there is ammonium production. There is a static drop of medium in polystyrene culture dishes are non-physiological, and ammonium production is there. Ammonium production can be by two things. What? Metabolism of embryo. Ammonium production can be by, first is embryo metabolism. Embryo metabolism. In this, 25 picomole per embryo per hour of ammonia is formed. And due to spontaneous amino acid breakdown at 37 degrees Celsius. These two contribute to the ammonium production. Okay. Now, impact on embryo. What is the impact of embryo development in ammonia, more ammonia? If ammonium concentration is more than 300 micromole, okay, then first of all, it will affect the embryo development and differentiation. Second, it will affect the fetal growth rates and normality. Third is it will negatively impact the embryo metabolism, pH regulation, and gene expression. Migrating ammonium toxicity. Now, mitigating, how to decrease ammonium toxicity. We can decrease ammonium toxicity by replacing culture media every 48 hours. Secondly, we can replace it by labile glutamine. We can replace the labile glutamine with the stable dipeptide form, that is alanine glutamine. What is the advantage is this alanine glutamine will reduce the ammonium level and it will enhance the embryo viability. Okay. Now see, there are two media, KSOM media and G1, G2 media. Okay. We can see in the diagram, this is the KSOM media where ammonium is accumulated. Okay. See, but in this G1, G2 media, ammonium was unable to accumulate because in this we used alanine glutamine, which is a stable form of the dipeptide form of the glutamine, stable dipeptide form of the glutamine, which result in low ammonium level. Okay. Now see, we can see in this bar graph that impact of ammonia on embryo development. Day 3 embryo development is also decreased by ammonia, and total blastocyst development is also decreased by the ammonia. See, this shows the pyruvate uptake by embryo at various post-thaw time points. The pyruvate uptake. Now the red bars is the presence of embryo. Pyruvate uptake is low in the presence of ammonia. Higher pyruvate uptake if there is no accumulation of ammonia. So ammonium exposure impairs embryo metabolism as indicated by reduced pyruvate uptake. See, it talks about the gene expression. Ammonia alter the gene expression in human blastocyst with clear differences between the control and the exposed group. Now we'll talk about the sequential media and monoculture in the next class. We'll talk about it in our next class.